All right, let's get going. Now, to this audience, this is probably a stupid slide to present. This is the most profound thing I can tell you tonight, or this morning. This is why we are all here. In many ways, think about it. But if you ask an evolutionary developmental biologist like me, what's the meaning of life? It's to pass on your genome so that it gets inherited by future generations and your lineage is complete. And a big concern then, these are my F1s for example, we want our kids to be able to pass on our genes to future generations. And that's what I mean when I talk about hybrid developmental dysfunction. When I talk about hybrids, I'm not talking about hybrids between species, I'm talking about hybrids that are combinations of genetic material from two distinct individuals of the same species, and how, hopefully, my wife and I did not pass through sperm and egg bad combinations of genes into our two daughters that will cause them to have developmental problems. And I'd like to thank, first of all, the organizers for the opportunity to talk. And I'd like to mention that we've been tweeting with the # West SDB 17 for those of you that are on Twitter. Feel free to contact me. And by the way, I'm actually very impressed for a developmental biology crowd. You've been making your slides pretty colorblind friendly. Thank you for that. I've done that. So red and blue is coding. Now, Darwin was a huge proponent, of course, of understanding the origin of species and how genetic incompatibilities, like those between the horse and the donkey, if you mate them together, if you make a mule, the mule is sterile. That's the biological species concept of Ernst Mayr. And what's more important is that this is a developmental process. The formation of species, the divergence of one population into multiple species that are either having dead or infertile F1 hybrids is a developmental process, because death, hybrid development, when it doesn't work properly or when those individuals are infertile, those are both inherently developmental processes. So really, the origins of species should have been by means of hybrid developmental dysfunction, not by natural selection. Actually, both. Fair enough. So this is what I'm talking about. And instead of using horses and donkeys, because those are adaptable genetic organisms, I'm going to be using worms instead. So the question is, what genetic differences between two populations or two individuals of one species causes speciation? And more specifically, when I say speciation, I mean developmental dysfunction. Some variation in development causes reproductive or developmental problems. So I work on Caenorhabditis nematodes. You're probably all somewhat familiar, at least with C. elegans. A great developmental model system, I probably don't have to mention, but they're eutelic. Every individual has the exact same number of cells, and what's more useful for us in the laboratory is that Caenorhabditis, both elegans and briggsae, the species I work in, have self-fertile hermaphrodites. So a single hermaphrodite will produce somewhere in the range of 300 of her own oocytes, offspring, and each of those offspring will undergo a series of molts through 4 larval stages until becoming a self-reproductive hermaphrodite again. And this is really important for studying what happens in nature in C. briggsae, the species I work in. So Caenorhabditis briggsae is a really close relative of C. elegans. And the reason we use it is because it's much more genetically diverse in an interesting way than C. elegans populations. Geneticists and molecular biologists and ecologists have traveled around the world collecting these natural isolates, we call wild isolates or wild strains. And this is just a phylogenetic tree representing the genetic diversity among C. briggsae genetic isolates. And what was striking is that there is a group that are tropical, and they're all most closely related to each other. And then there's a temperate clade, where those individuals are most genetically identical to each other. And because these worms primarily exist as hermaphrodites in nature, there's something like 1 in 1,000 individuals as male. Lucky for the male, I guess. Most of these worms are selfing. They're self-fertile. And that means that in nature, if you find an individual, if you look at its cell and chromosomes, like the mitochondrial genome, circular, and an autosomal pair, these are diploid species, they're going to be completely homozygous throughout their entire nuclear genome because they've been selfing. There's no genetic variation within each of these populations. So I don't know if Mrs. or Mr. works for hermaphrodites, so I've called them MX. Mrs. Scarlet and Mrs. Peacock. I'll be color coding these worms as blue and red for temperate and tropical climates respectively. And what I want to do then is look at hybrids between these members of the same species. They're fertile when you cross them. They're about 1% genetically divergent on average, so about 1 in every 100 nucleotides differs. Is that enough to cause some hybrid developmental dysfunction? And it turns out yes, and a collaborator of ours, Scott Baird, originally noticed this. When you cross Miss Scarlet and Miss Peacock, the red strain and the blue strain, about 20% of their F2 hybrids become developmentally delayed. What this is a micrograph of is a wild-type F2 individual and its age-matched delayed sibling. So these were born, hatched at the exact same time. About 3 days later, the wild-type sibling is fully reproductively fertile. It's an adult, but its poor sibling has undergone some sort of developmental delay and is still a larva. The reason this is a problem for health and for fitness and for evolution is because the person, or the worm, that gets to the resources first and starts reproducing and letting their kids have access to, in this case, the bacteria that these worms eat, has an evolutionary advantage. They have more kids, and they're better able to pass their genome on to the future generations. So that means that this is indeed a developmental issue that causes a long-term problem in terms of speciation. So it's much like genetic differences between the red and the blue strains of worms, in this case, are causing some sort of genetic incompatibility that slows development in a fraction of these hybrids. So this sets up, it's 20% of those F2 offspring that are delayed, by the way. It's an interesting percentage, 20%. So a model might be that you have a measure of fitness in terms of development rate. So normal development would be high fitness. We know that the P0 generation worm, the blue worm, has a totally blue homozygous genotype throughout chromosome 3 in this case. It turns out that the delayed worms all have red alleles, homozygosity, on chromosome 3. So those worms that inherit both chromosome 3 alleles as being from the red parent get developmental delay. The weird thing is that that's the same genotype of the red parent. Totally homozygous throughout the entire genome, right, up here. This worm is fit. This worm has delayed development. So the question is, what's the interaction between being homozygous for red alleles on chromosome 3? Is there a dominant or a recessive interaction between those red alleles on chromosome 3 and blue alleles, blue parent genotypes, elsewhere in the genome? The chromosome, we don't know what it is yet. And punchline, we don't know yet. So by the time we get to the end, I won't be telling you what the gene is or necessarily which chromosome that interacting locus is on. So this is a complex trait. It's got at least 2 loci involved in genes coming together from 2 different parents into one offspring, causing them to be developmentally delayed. We're trying to figure out what is that gene. And we're attempting this in a couple of different ways. We've been using near-isogenic lines of briggsae to try to map this chromosome 3 locus to identify the gene that's on chromosome 3 that causes developmental delay, and also doing some bulk segregant analysis. And I'm happy, unfortunately, for the CSU system, those of us that are on semester systems, this is not our spring break, which is why I'm here talking on behalf of many of my students who I'd rather have here doing this, because it's their work. But I'll be summarizing their work here. And then we're also going to be looking here at the bottom to try to better define this phenotype. All we know right now is that it takes the delayed worms an extra day to reach adulthood. But we don't know when that starts and when that period stops. I'm trying to do this for QTL mapping later. Please do not concern yourselves with the details, because I know you can't see them. But at the top is our F2 bulk segregate analysis, which was done by Leonardo Velasco, a former undergraduate student in my lab, who took individual delayed worms and genotyped them across chromosome 3 here on the x-axis. And we found a region of peak association where we're measuring on the y-axis the percent of all of the delayed worms that had homozygous genotypes with that red allele. So we expect that when we find the region of the genome that contains the gene on chromosome 3 that's associated with this trait, we would see 100% of delayed worms having that red homozygous genotype. And there are three, take-home message, there are three at least interesting candidate genes in there that you can't see. UCR1, MEV1, and MRPL22, which I'll come back to in a few minutes, are mitochondria-related proteins. And using a near isogenic line approach, former graduate student Jordan Montgomery made a series of near isogenic lines that had AF16 genomic DNA in the middle of chromosome 3 introgressed into the blue, so the red alleles in the middle of chromosome 3, in the blue nuclear background. Everything else was blue in the genome except for a tiny little fragment of chromosome 3, and that genome segment matches what we found in the F2 bulk segregate analysis. So it looks like this is a defined region of chromosome 3 where one of the genes is that's responsible for hybrid developmental dysfunction. It may be one of those three candidate genes. And I also mentioned we wanted to look at when exactly does this process of developmental delay start and stop during development, which takes about 72 hours from birth to adulthood, as I said earlier, in Cybrids and in C. elegans. And one of my technicians and an undergraduate student, Leslie, had taken a series. This is just an example, and again, you're not meant to see details here. We took individual micrographs of worms over large timescales with lots of worms comprising both F2 wild-type worms, their delayed siblings, and the parents. This was the red strain, and this is the blue strain, and compared length of development. Here's maybe more useful blow-up image of a few of these individuals. And we found that there was no significant difference at 24 hours post-hatching between these four groups, but hopefully even from the back of the room you'll be able to appreciate that the F2 delayed worms are much smaller in length at 48 hours compared to the wild-type worms and their parents, and by 72 hours these worms have caught up, the delayed worms have caught up in development. They're no longer statistically differently sized from their wild-type siblings or the parents. So the onset of delay is somewhere between 24 hours, and the exit from developmental delay occurs around 72 hours. The title of the talk has something to do with mitochondrial and nuclear, and this is where this process comes in. We created a series of cytoplasmic nuclear hybrids, which are called Cybrids, and this is complicated but simple. So here's the simple version. You take the mitochondrial genome of one strain and you put it in the nuclear background of the other strain. And this tests for whether or not there are genetic interactions between mitochondrial genes and nuclear genes that cause a particular phenotype. And we would predict, and I'm going to show on the next slide, we have data that fitness, when measured in the number of offspring produced, when you have the parent genotypes, pure blue genomes or pure red genomes, you get high fecundity. Lots of kids. But when you mismatch the mitochondrial nuclear genomes, you get a decrease in fitness. And that's work that's already been published. So we have the pure red genome here with having about 225 offspring per hermaphrodite. But when you replace their red mitochondrial DNA with the blue mitochondrial DNA here, you get a significant reduction in the number of individuals produced. And again, this is a developmental issue because the number of offspring that a hermaphrodite can have depends on how many times her germline stem cell divided to provide her with her self-sperm. One of the reasons that this might be the case is mitochondrial nuclear coevolution. If you weren't familiar, mitochondria and nuclear genomes both encode molecules and proteins that are involved in electron transport. So the blue ones here are nuclear encoded subunits of the electron transport chain, and red ones are ones encoded by the mitochondrial genome. And so there's precedent for the possibility that what's happening here is that there are genetic differences between the red strain and the blue strain of these worms. And what that does is create, when we make a Cybrid, for example, or even an F2 hybrid, we create complexes of proteins, say, that don't quite work well together for steric or other reasons. And these could be, in my case, electron transport chain components. So it could be that there's mitochondrial dysfunction in these organisms, and that's what's causing them to have slower development and or fewer offspring. Or, to the rest of you, because I haven't shown you any micrographs of cells or signaling pathways yet, this could be signaling pathway molecules. It could be divergence in the ability for signaling to occur. And we also then, given this hypothesis that maybe mitochondrial nuclear genetic interactions are causing developmental delay, we thought, well, we already have our Cybrids in the lab, we better measure their development rates and see if they're changing or not. And indeed, at 48 hours post-hatching, the Cybrids here are all plotted in dashed lines, and the pure strains in the solid lines. There's a group, 4 out of our 5 replicate Cybrid lines with the mismatched mitochondria are significantly slower to develop in terms of length, total length of the worm. So it looks like there's a mitochondrial nuclear co-evolutionary separation when we make hybrids or Cybrids, the developmental delay. So in general, this is the summary. Mitochondrial nuclear interactions seem to extend larval development, decrease self-fecundity, and I'm assuming, we haven't tested this yet by oogenesis, but that's our top pick. And then this maps to a region of chromosome 3 that, remember, contains some mitochondrial genes. So part of this interaction between chromosome 3 and some unknown chromosome, which we'll get to, might have to do with mitochondrial nuclear protein actions. One last point before I give up the mic, thank you very much for your attention, by the way, is we've got some really interesting results from our lab based mainly on a project from Shadi Adena, who was a former graduate student in my lab, that shows that in Cybrids, when you hybridize species populations like Miss Scarlet and Mrs. Peacock, mitochondrial DNA gets in from the dad sometimes. So all of that stuff you learned in the textbooks about you only get your mitochondria from your mom is not true. Not always true. So my caution is, again, to try to make this relevant to as many of you as possible, especially you in the back of the room. If you're doing anything in biology that involves crosses, not something that's asexual, like tissue culture or using asexually reproducing organisms, if you're using any sort of cross and you see any sort of developmental defect, think, could there be some paternal mitochondrial leakage? That is, could some of dad's mitochondria be getting into offspring? And could that be at least partly causing the effect that I'm seeing? So with that, I've got to thank some more. Pictured are students who are also parts of different projects that I haven't had time to mention. But thank you very much for your time. Thanks to the funding sources, and I'll take questions if there's time.
Time for a couple of questions.
I noticed there was one mitochondrial hybrid strain that had no differences in either growth rate or offspring. Have you done differential mitochondrial genome sequencing to see if there's any loss or non-loss of genes?
Fantastic. So there was one Cybrid that didn't behave like the rest. There always is, one of our replicates. And that is one of the things we'd love to do, is to figure out what's genetically different about that strain. There might be some compensatory mutations, for example, in the nuclear genome or the mitochondrial genome that has restored fitness in that strain. And right now, we're working on not only doing that deep sequencing, but also creating some long-term evolution experiments to look at genetic changes in the nuclear and mitochondrial genomes that allow those Cybrids to regain fitness. Thank you.
If there aren't any other questions, then we'll stay on track for lunch.